Go ahead.
Let's do it, guys.
Hello.
Good morning, everybody.
Good
First
morning.
of all, thank you very much to join us in this webinar, which we are going to talk about the requirements for all countries that request QSAR studies. It's a pleasure to have you here with us, and I expect you to learn a lot about what we are going to say here. Okay? We are still accepting some people which join us here. Happily, I want to announce you that we have like 9 people subscribed in this webinar. That's a wonderful amount of people, right? So thank you very much to trust on Smart Talks, on Bioacting and LASA. Right, we still have some people getting. Just wait for some 2 or 1 minute to admit all in order to don't lose any information. Let's start here. Presentation, I will share my screen. I suppose everybody can see my screen. Okay, let's wait just one more minute, and then we start. Okay.
Buenos días.
It's 8:03, so let's start. There is some people still join us, but as they are requesting to get in the room, I will admit them in the webinar. So again, thank you everybody to join us in this webinar, which we are going to talk about requirements for all countries that request QSAR studies. And before that, I would like to say to you how the webinar will be conducted, right? So the webinar will take like 1 hour duration. We have 2 speakers, right? One is Pedro, which will talk about how the QSAR is requested in several countries, how it works, and how the QSAR is elaborated, right? And for different countries. And afterwards, we have Raisa, which is representing LASA, right? The software which performs the QSAR evaluation, right? So that will take around 40 minutes, both presentations. And then we are going to have the Q&A sections, okay? The Q&A sections will be made in 2 steps, right? So the first Q&A section will be made by chat. All of them will be made by chat, but some of the questions will be answered, right, through the chat by Hernan, which I will present afterwards to you. When you want the question be answered by one specific speaker, please describe the speaker that you want to answer that question. And at the end of this speech of this speaker, we are going to address this question to him. Okay, so that's how this webinar will work. If you have any questions, please describe or write in the chat in order to address to you. Okay. Before we start the webinar, I want to introduce Smart Talks to you. So Smart Talks is a company which has grown in 2017, right? With a previous experience of 20 years in the agro market, okay? So we offer smart solutions to the agro industry, working as a hub of agro solutions. So we work gathering the best professionals in the market to attend clients which are abroad, Brazil and Latam, to solve their issues and bring solutions to them. We attend the national and international market with a multidisciplinary team, right, using national and international partners. Okay, so that I'm going to show you in the next slides. For in Brazil, we offer these services, which is the QSAR and SAR for Brazil, Europe, Mexico, for regulatory and exploratory purpose. Okay, so that's the countries which request the predictive toxicology evaluation. Also, we provide the risk assessment, which is focused on Brazilian requirements, right? Also, authorities' requirements and technical advices when the companies has some issues with the dossier, we can support them with the lawsuit support too. Okay, so we also do the toxicology evaluation, ecotoxicology evaluation. We implement, sorry for that, this is still in Portuguese here, right? But I will translate to you. We implement, monitor, and evaluate 5 batch studies, right? And also we elaborate the PATI, which is the technical report which Anvisa's back in Brazil requests for some companies, right? And also the new tool that Anvisa is elaborating for some companies, which needs to fulfill this spreadsheet. So we support them with this services too. For registration, we have partners all over Latin America, right? So we have partners Argentina, Paraguay, Uruguay, Peru, Bolivia, Chile, Ecuador, Mexico, Colombia, as well as Brazil to make the registrations for pesticides, for food additive, for biological products, and also do market evaluation for all those countries, okay? We expanded like 1 year ago for Canada too, right? And US. And let's make a brief introduction about bioacting. That will be just a brief introduction that will be deeply exposed and explained by Pedro afterwards. Okay, so bioacting, a company founded in 2012, located in Buenos Aires, Argentina. Since the beginning, they have represented a large number of clients globally. Pedro has a well-known network around the world. Sorry for stopping talking because I'm admitting people here in the webinar. So they have provided innovative solutions to problems generated in the field of regulatory and develop a sustainable products. Okay, so bioacting offers to the market in the regulatory area, formulation, support area, training, predictive toxicology, which we do in partnership with bioacting, experience and emblematic cases which happens, and Pedro will explain better to you, and as well as 5 batch training and implementation, which we offer together with bioacting, with the expertise of Pedro and Hernan that I will present afterwards to you. And LASA which is the company which provides the software, the expert system to make all this robust evaluation, was established in 1983. The headquarters is located in the United Kingdom as a not-for-profit and education charity and creator of knowledge base, statistics and database system, and facilitate collaborative data sharing projects and chemistry-related industry. So that's just a brief explanation that Raisa, which is representing LASA, you will deeply inform you about LASA with more property. Okay, so those are the expert systems that LASA offers that the focus of this webinar will be direct nexus that we use for agrochemical purpose. Okay, so that is very much that I want to tell you. And now I will introduce to you Hernan, which is our specialist, right? So the specialist of QSAR evaluation, and he will be the one which will be answering your questions through the chat, right? So if you have any doubts during the webinar, you can write it in a chat, and then will answer to you. As I said, if you want to have a specific speaker to answer your question, please describe the speaker that you want to answer that question, right? So Hernan is a master's degree in organic chemistry by Universidad of Buenos Aires and works in agrochemical field since 2006. He has a vast experience in global equivalence registration process, physicochemical 5-batch analysis toxicology, including silico, ecotoxicology studies, being the last two since 2013. Currently works in a GLP lab in Argentina as a study director, at Bioactin and at Bioactin as a scientific director since 2012. And as our speaker, which is Pedro Guvidalves, he is a PhD organic chemistry by the City University of New York, which elaborated the thesis in enantiomeric synthesis of biological active phospholipids. Also, he has a postdoctorate in medical chemistry at the University of British Columbia. He has more than 15 research publications in internationally recognized journals and 30 years of experience in agrochemicals for product process research and development. Was a speaker in several congresses like CRAC China, CAC China, American Chemical Society, and ENIGMA. He will speak today about the QSAR requirements and all different countries which request such studies. So Pedro, thank you to join us in this webinar, and the word is yours to give all this knowledge that you have to us. Thank you very much.
Oh, okay. Thank you, Alexandri, for the presentation on Bioactin and myself. Well, if you can start showing my presentation, it will be great. We're going to talk about predictive toxicology, which is in silico analysis. And it's a partnership that we have with SmartTox in Brazil. And we're going to go to the next slide, please. The presentation diagram is we're going to talk about the definitions of in silico, which is SAR and QSAR, requirements in different countries, the different softwares, the global tendency, and finally the group. What is important here is to understand a little bit about a little bit of a background. When you have a new product, is the first registration, and it's the full registration. Most of those products obviously have patents. The full package is related to all the ectox and ecotox studies that you need to perform, and that's the reference source, which is not mostly the case of the people that are attending this webinar. But when there is a product already registered, you can go through the registration process by equivalence, which you will have the reference source, obviously, as the reference, and you need to conduct several studies, physical chemical studies, and especially 5H analysis, and just the acute toxicology, the 6-pack, which is called the 6-pack plus mutagenesis and an M test. Okay. Well, next slide, please. When you have an active ingredient, the importance of the active ingredient is the composition, and the composition comes from the 5H analysis. Okay, and obviously also the toxicology. When you have the same chemistry, and it says that you have the same toxicology. So, and that's the base on the equivalence registration: same chemistry, same toxicology. And the composition is determined, that equivalence is determined by the reference. The reference has already published in the different countries to the different authorities what is the profile of its active ingredient. And some of the impurities and the profile that you have are relevant impurities which are determined by FAO, EFSA, EPA, etc. They are published by them, and those are relevant impurities, and they are well described in those different institutions. Okay, in Brazil is the ENS 02. And the toxicology is also well known. It's published in the different and defined by the different also institutions like FAO, EFSA, EPA, etc. If the composition is not equivalent, it's needed to you need to prove the difference that has no impact in the toxicological profile, and that's why where we enter some studies like the QSAR analysis, which is very important. So we have, as we said, next slide, please. Next slide. We have, as we said, chemical equivalence, which is based on 5H analysis, quality and quantitative declaration, Phys-Chem studies like melting point, low point, vapor pressure, color, or solubility in water and in organic solvents. Those are basically the studies that you need for chemical equivalence. Then you have toxicology equivalence, which are the acute reports, which are acute oral, dermal, inhalatory, skin sensitization, Ames test, mutagenesis, etc. Some countries use those tiers. The tier 1 is chemical equivalence. If you are chemically equivalent to the source of the reference, then you pass the tier 1. As we said, the same chemistry, the same toxicology. If it just happens that you don't have a chemical equivalence by your profile against the profile of the reference, then you need to go to tier 2. In order to go to the tier 2, you have to pass through the QSAR analysis, which is quantitative structure activity relationship. And as it says very clearly, it's quantitative, and there is a relationship between the structure and the activity of that impurity that is not in the reference or is higher or a particular impurity that is in the reference but is in higher concentration, whether it's 50% or 3 grams per kilo, whichever is the most restricted one. And then you have to go through the acute toxicology. And we need to say that the QSAR analysis gives you the chance not to use animals, which is a tendency in the world. Okay, next slide, please. As we said, in Brazil and in different countries, if the profile is chemically equivalent, the phase 2 is not needed. As we said before, phase 1 is the chemical equivalence. But if the profile is not chemically equivalent, you have to go to phase 2. Europe has the same system. When you have to go to the phase 2, you need to perform a QSAR analysis on the impurities that are not in the reference or are in higher concentration than the reference. And then afterwards, some acute toxicology could be asked from the authorities to be performed. What is a QSAR analysis? The QSAR analysis are capable to find a relationship between the structure, some fragments or descriptors through the LOPO, and relate that to the biological activity. When the product is not equivalent with the reference, 2 options are possible. As we said before, there could be a presence of a new impurity that is not described in the reference, or one impurity that is higher than the reference. That is, a reference could have described that impurity, but in your case, that impurity is in higher concentration, whether it's 50% or 3 grams per kilo, whichever is the most restricted one. For both cases, QSAR studies for those type of impurities need to be performed and need to be proved that the presence of these impurities do not pose in your technical material, that does not generate any increment in the toxicology behavior of your matrix. Your matrix is your product. That's the basic of these rules. Okay. I will give you some. Next slide, please. I will give you some overview on how the QSAR analysis is structured. In the 1960s, Hans and Fujita proposed a method to describe in qualitative form the relationship between the biological activity of a chemical and the descriptors. And there is a linear correlation of free energy between them. And the biological activity is expressed by F, which are the molecular descriptors of the fragment or the alerts, as we said, that appears in the molecule. There are some descriptors or fragments or alerts that gives you some kind of a alert in terms of its potential potential to be some kind of that will pose some kind of toxicology alert. In the beginning, they used a descriptor. The descriptor that was used was lipophilicity, considering that generally they could obtain a parabolic relationship between the partition coefficient, or what is called lipophilicity, and the biological activity of an homologous series of compounds. And that's what Raisa will explain a little bit better how it works. And there you see the formula, which uses the inverse of the concentration that generates a some type of response due to that lipophilicity, whether it's LD50, EC50, etc. Okay. Next slide, please. So in the QSAR analysis, we have a central axiom, which is basically that similar molecule will behave similarly. Okay. The activity, which are the, as we said, the molecular descriptors, and three principal components. One is a dependent variable. It's a data set that provides activity for a group of chemical products. Okay, group of chemical products, and the data sets of properties related to the structure for those for that group. Okay, and those are independent variables of the related properties, and the model will relate those two variables, the dependent variable and independent variable. One model relates to both data sets. And the fragment identification, which is very important, which are the alerts that are associated with certain biological activity of those alerts. For example, polychlorinated compounds, polyoxygenated compounds, nitro compounds, amine compounds, whether are primary, secondary, or tertiary. So this fragment identification is quite important, and that will appear in the which is called the SAR analysis, which is previous to the QSAR analysis. SAR analysis is just structure, activity, relationship. And most platforms use several algorithms which are necessary to perform all the calculations for those predictions. And the different platforms have different algorithms. Even within a platform, you may have different models that uses several algorithms. Okay, next slide, please. How do I obtain the homologous series? And you have how all the platforms obtain those homologous series. The structure is analyzed in a function of fragments associated with certain toxicology, and those are the alerts, as we said before. The alerts will be used to categorize that database. As we said, those alerts are of certain fragments are known. For example, fluorinated compounds, chlorinated compounds, nitro compounds, amine compounds, which will pose certain alerts. Okay, based on the results, those compounds where the alert is present are with high similar structure are chosen. You choose those compounds that have similar alerts, and once the database is chosen, you have a graph which is made of the activity and the physical chemical properties. And how do you obtain those physical contents, physical chemical constants? Those constants are made through algorithm, and then those algorithms will estimate those physical chemical constants. When you have both of them, the estimated physical chemical constant is extrapolated in order to give a toxicological result. So with those based, then you perform your database. With this analysis, we can estimate the physical chemical constant and obtain a toxicology information. And that's a great tool because, as we said before, you don't use animals. I will give you some examples on the alerts. Next slide, please. And here we have depicted in red, which is a cyano compound. This cyano compound, the hydrogen is very acidic because of the presence of this CN group. And this alert will have a possible protein binding, okay, OASIS or ECD, but also a nucleophilic attack. It's an SN2 reaction. An SN2 reaction is a reaction where both components are important in their concentration. Okay, it's a substitution nucleophilic reaction, which is called SN2. And there you can have the reaction of an amine group, which could be present and are present actually in the DNA base. Since the DNA gene and the DNA base has amine compounds, you can have an SN2 reaction and you compose a change in the DNA, and then you can have a gene aberration, and that will lead to genotoxicity. Okay, chemical structure with such reactivity, which SN2 electrophiles, because the carbon attack could be, for example, benzylic at the benzylic position, like in this case of the CN, or you can have an aromatic compounds with nitro groups, which are electron withdrawing, and you have if you have in the molecule a chlorine group, that chlorine group could be attacked and displaced. So you can have a possibility of a DNA aberration. And also, what is important here, for example, in the other structure in red, there you can have monoalkenes, okay, where you have there a double bond, a CF3 group, a chlorine group, and a methyl group. This could have a high gene expression, as is depicted there also in red. What happens here is that you can form a positive charge, as we can see there, and the metals in our body, like iron, could form an epoxide. And an epoxide is very reactive. That epoxide could be attacked by the DNA. But we need to see if that positive charge is possible to be formed, whether the groups associated with that positive charge are electron withdrawing or are or give electrons. We could stabilize or destabilize that positive charge. So it's very important here, the chemistry, because not only QSAR analysis is important, obviously, but to have a critical analysis in terms of the chemistry of that particular impurity or that particular fragment. Because in this case, which is shown there, that alert, which is in red, we can clearly see that there are two electron withdrawing groups, the CF3 and the chlorine group. And that could destabilize that positive charge. Therefore, it would be very impossible for any attack or any formation of an epoxide, because it's very the equilibrium of that particular oxygenation by cytochrome 450, it will not be possible. Okay, so the critical analysis in terms of organic chemistry is also very important in a QSAR analysis. Being Bioacting a multidisciplinary company, we have experts in chemistry also, in order to justify and do a critical analysis and a mechanism of certain reaction that may pose a gene aberration or attack by DNA or interaction with a protein. There are other problems like steric hindrance, which we'll show in the next slide. Okay, next slide, please. In this case, we are talking about fomesafen, for example. You can see that it's a nitro group ortho to one of the group, and there is a CF3. It's important also to understand that whenever you determine an impurity in your compound, it's very important to give the right structure, because if you give the wrong structure, a nitro group in a different position could have a different impact, like in impurity 1 and impurity 2. One is in a different position that in fomesafen, and in the other one is a nitro group in the other benzene ring. Okay. So there is a possibility of DNA binding of that nitro group, and you can see in impurity 1 is too crowded. There are so many substituents in one of the benzene ring that it's almost impossible because it's very crowded. In impurity 2, being the nitro group in the other ring, you can see that is the crowdiness is less. So it's important to understand also where you put your nitro group, and which compound is more toxic and why. And as we said before, the impurity 1 is less toxic than fomesafen, and fomesafen and impurity 2 is almost the same. Next slide, please. Well, which compound is more toxic? As we have two possibilities: polynitro, polynitro, because as you can see in the impurity 3, you have the two nitro groups in the same position, and in impurity 2, as we said, you have the nitro group in the other ring. So here clearly a polynitro compound in the same benzene ring is quite much more toxic than the other one. You can see the LD50 in red is almost 10 to 12 more toxic, 1,400 milligrams per kilo against 125. Because the polynitro compounds obviously makes the benzene ring extremely exposed to substitution, because both nitro groups will push a lot of electrons and will make that benzene ring very exposed to SN2 reaction or any ring substitution reaction. And that ring substitution reaction could lead to gene aberration. This evidence is very important on the Alex and the correlation election of the homologous series. Even if the structural similarity is high, the toxicology is not comparable since the alert presence are different. Okay, so not only the position, but also how you're gonna relate that to the homologous series. Okay, I think we have you can go to the next slide. We don't have much time, so I will, as I said before, the alert is the polynitrated compounds interaction with DNA. Okay, so it's extremely reactive, as we said. So that impurity is much more toxic than the other one. Well, next slide, please. Well, the software are statistical-based model. They are mathematical models, and we have SARA Nexus, which Raisa will talk about, and is one of our partners, and Derek Nexus. Also, we use a lot of them. We believe that Derek is a very strong platform and give us a lot of insights on the QSAR analysis. We have Case Ultra, Vega, EPI, etc. And the expert rule based on those models, and those are knowledge models. We have Derek's, QSAR, Toolbox, ToxTree, Vega Hub. Basically, those are the software. And the software versus the models. Because different platforms and different expert systems have different models in order to calculate the quantitative structure activity relationship. And we have Direct Nexus, QSAR Toolbox, Vega Hub, and Test EPA. And those are the software. But the models, for example, for Test EPA, you have different models: consensus, a right-hand clustering, a single model, nearest neighbor. So you have different models. So not only it's important the software or the platform, but also the models that you can use, and different models could have different results. And then that's where it comes the critical analysis. Okay, and the false positive and the false negatives. Okay. Next slide, please. Next slide. Next. Okay. Different types of approaches that you have, as we said before, especially Europe uses this approach, which first you go through the SAR analysis, the structure-activity relationship. That's a qualitative approach, okay? That qualitative approach detects the alerts, the descriptors, the fragments, okay? And doesn't give you any results, it just gives you the alert. Only potentially based alerts that may pose a risk. The presence of alert is not enough evidence to assume that this structure is toxic, but yes, it gives you an alert. Then you go to the quantitative approach, which is the QSAR analysis, which is the quantitative structure activity relationship. That quantitative structure activity relationship detects the alerts, the descriptors, and the fragment, but will give you a result, will give you a quantitative result, and a correlation between the phys-chem properties and the function and their function and the biological activity. Okay, so this is the case in Europe, and this will be, I guess, in place in India soon. And this is the strategy of Europe. First, go through the SAR analysis. If there are no alerts, then you don't go to the endpoints. But if you have a SAR, then you have to go to the quantitative approach. If a certain alert gives you a possible genotoxic, then you have to go to the quantitative approach to that particular alert. Okay, international requirements. Go to the next slide, please. Even though we are in Europe, France and Netherlands have, I mean, the QSAR analysis actually is it's an umbrella, okay? But many different countries could interpret or could go through your study in different ways. According to France, for example, not a specific QSAR model is required, okay? But yes, two independent, reliable QSAR models, okay? Okay, they don't still tell you which platform you may use, but you have to use two reliable ones, okay? And in the Netherlands, the SAR analysis must be performed using two recognized systems like Direct Nexus and QSAR toolbox. So as you can see, different countries may have a different approach, but at the end, SAR quasar analysis is important whether you use one platform or two platforms or the SAR analysis or not. In our particular opinion, you need to use different models. I'm sorry. You have to use different different platforms or different expert systems with different models in order to make the study more robust and could give you different insights, and with the results and your critical analysis in terms of organic chemistry, reaction mechanism, or seeing the structures as we showed before, that could give you a better insight and a more robust report, justifying everything. So in our opinion, a full report with different models, with different platforms and different models, whether it's SAR or quasar analysis, is the best option. Okay. In Australia, two types of in silico methodologies, that is two different types of platform must be used: an expert rule-based model and a statistically based model. If one model shows positive result, a genotoxic testing is required. See here the importance of having a very profound knowledge, not only on toxicology, but also in chemistry. Chemistry is so important because chemistry is everything. You can justify everything through mechanism, through structure. See that even different countries has different views. You need to have a very strong study in order to cover most of the countries that you are interested in. In Mexico, it's a special case. They only need the LD50 endpoint, a quantitative number that will give you the platform. The worst case possible contribution must calculate using the FAO manual. Other endpoints can be required, but it's not usual. It depends on the authority, it depends on the results you get, or if it's a real structure that has some certain alerts that may give the authorities the right to request other endpoints. Okay, now we go to Brazil. Brazil, you need to perform all the endpoints independently. If the SAR analysis gives you a positive or a negative alert, you have to go through all the endpoints: acute oral toxicity, skin sensitization, mutagenicity, carcinogenicity, developmental and neurotoxicity must be covered to satisfy the legislation. And they ask for at least 3 specialist systems. You need to present the 3 specialist systems. And for example, direct nexus, test EPA, QSAR toolbox. But Brazil authorities look at the critical analysis very deeply. You need to submit that critical analysis for all endpoints and for all the systems. But they talk about only platforms. They don't talk about models. As we said before, QSAR toolbox has 5, 6, 7 different models. So Brazil stressed to use 3 different platforms. They don't stress to use different models. We as Bioactive and as a responsible company suggest to use different models, and that's a full analysis in order to be safe and to have a very robust study. But there are certain clients or certain companies that only wants to do 3 platforms and don't use different models. In some cases in Brazil also, a analog must be presented. What is an analog? A similar structure or a similar mode of action. For example, strobilurins. If you have impurities on your strobilurins, you need to compare that with your active ingredients, but you also can relate to other strobilurins because they have the same mode of action. In India, the guideline is under revision. Actually, it's a draft version of the QSAR analysis required for different impurities. The tendency is to use the same approach that the EU uses. That is, in silico analysis will be mandatory. SAR analysis, a structure activity relationship analysis, must be performed. If the SAR analysis shows any toxicant results or any toxicant possible alert, a QSAR analysis must be done in that particular alert for that particular endpoint. For the moment, we don't know. There is no specific QSAR model or QSAR platform that is required to use, but we expect that Derek from LASA and Toolbox from OECD will be two of the ones chosen. We don't know if it's going to be 2, 3, or only one. As I said before, we recommend always to use more than one platform. Next slide, please. Well, our expertise, we have expertise in Europe. We have presented a lot of QSAR analysis and SAR analysis for Europe and for Mexico and for Brazil. Altogether, we have more than 150 studies presented in the different countries with the different formats that Europe may require, the different formats that Mexico will require. As we said, in Mexico, it's only LD50, but authorities could ask you for more endpoints to be performed. In Brazil, SAR analysis, QSAR analysis must be performed for relevant endpoints using 3 different specialist systems, as we said. So, and in Brazil, especially, a deep analysis must be done, and I think this could be done for all the countries, especially in Europe, and in the future, probably India too. So Bioactive is capable of performing any of the requirements of any country. In our opinion, as I said before, we choose and we elect and we recommend to use, to do a study, a complete study, okay, with different platforms, different models, with a critical analysis, mechanism, applicability of the domain, presence of false positive or negative, etc. So in this case, you have a very robust study which will be accepted everywhere. Next slide, please. Global tendency. The global tendency is to use in silico analysis. Is important. Is an important tendency. Even if they are not mandatory in all countries, they are accepted in Argentina, Chile, Paraguay, although there is no legislation in Argentina, for example, yet. Some countries are changing the current guidelines to require QSAR analysis, for example, India, as we said before. Also, this type of analysis are used in pharma, in veterinary, in cosmetic, etc. So this is very broad. This is not only for agrochemicals. It's used in pharma, it's used in cosmetics. Found who are developing draft guidelines for in silico studies, and for example, skin sensitization. Maybe Raisa could give you some update on that. Could be performed just only with in silico analysis. It's been reviewed, and I think it's going to be in place in 2023. So in silico analysis is getting very important in the registration of agrochemicals, obviously, as we said before, pharma, vet, and cosmetic also. But in our sector is very, very important, and it's going to be very important in the future in order not to use animals. Okay, the last slide. As we said before, we have a triangle relationship between SmartTalk, Bioacting, and LASA. With LASA Limited, we have a strong relationship for many, many years. We believe the QSAR platform of LASA is very robust and very strong. Thank you very much for your attention. If there are any questions, we are happy to answer.
Thank you very much, Pedro. It's a lot of information to speak in like 20 minutes, 30 minutes. So it's a lot of knowledge. Thank you for sharing that with us. And now we are going to have Raisa. So Raisa is a business development manager at LASA Limited company. She studied chemistry at the University of Huddersfield in UK. After graduating with her master's, she joined LASA Limited 2.5 years ago, and she will be presenting and demonstrating today a LASA Limited toxicity prediction software by Derek Nexus. All right, so Raisa, the word is yours. Have a good presentation and share all your knowledge with us. Thank you.
Thank you very much. Thank you everybody for joining us today. As Alexandra mentioned, I'll be sharing a little bit about Derek Nexus, which is LASA Limited software. So I will share my screen. My agenda today, I know that we are slightly low on time, so I'll try to be quick. I'm going to give you a quick introduction to LASA Limited, which is the company that develops the software. I'll talk a little bit about the two approaches to toxicity predictions, and then I'll go into what the software is, how it works, and also give you a very quick demonstration. So this slide was already presented, but I wanted to touch upon a few points. LASA Limited is a not-for-profit organization and an educational charity. What this means is that any money that we make through the sponsorship of our software is invested directly back into our products and back into the company, which means that the software is constantly being updated and improved upon to make sure that we have the most up-to-date knowledge and it's validated and working well for our members and people who are using our software. And also the educational charity aspect, we have very close relationship with academic institutions around the world and with government regulators as well. Not only do we provide software for toxicity, which I'll be talking about today, we also have various other systems such as Zenith for degradation, Meteor for metabolism predictions, and Vitic, which is a toxicity database. If you're interested in any of these, you can go to our website to find more information, or you can put something in the chat and I will get back to you. If we're focusing on toxicity, there are two approaches that you can take. The first one, when you're looking at a system, is to take into consideration factors such as structure, but also activity, mechanism, metabolic data, and reactivity. A human expert, so a scientist, will gather all of this information together and write rules and develop structure-activity relationships. So this is the SAR approach that we talked about. It's an expert system, and LASA have Derek Nexus, which we provide to companies. On the other hand, it is also possible to have a system which just takes into consideration structure. So this uses an algorithm or machine learning. This will form hypotheses. And this is a QSAR methodology. And we call this also a statistical model. And at LASA Limited, we have Sarah Nexus. Now, both of these tools can be used together for various regulations around the world. As Pedro mentioned, you might need a SAR and a QSAR approach. For the purposes of this talk today, I will just be focusing on SAR and direct nexus because commonly that's the first step that you would take in your risk assessment. So as I mentioned, the software is a knowledge-based expert system. We have humans, scientists at LASA that are curating all of the data that they find online, publicly available data, even proprietary information that companies have donated to us. And they develop these structure-activity relationships. And from these, we cover 74 toxic endpoints in the software. So we have mutagenicity, carcinogenicity, skin sensitization, nephrotoxicity, hepatotoxicity, just to name a few. And not only do we give you a prediction to say if your compound is toxic or not for a specific endpoint, we also want to be as transparent as possible. So we will give supporting references and examples, which I'll show you in the demonstration. This is to help you with your regulatory submissions, so you know that you have all the information that you will need to be able to share the report with regulators at the final stage. Not only do we give positive predictions, so we will tell you whether your compound is positive for toxicity for 2 specific endpoints, so mutagenicity in vitro and skin sensitization. It's also available that we can tell you if your compound is negative, and we call these negative predictions. In terms of the methodology and how it works, it's a 2-step process. The first step is to identify any toxicophores in your compound. So you would put your compound into the software, and you can see this area highlighted. This functional group may be responsible for toxicity. So that would mean an alert would be fired. The second step is to make a qualitative prediction, and this takes into consideration various factors. So we will look at things like molecular weight. So you know that if your compound is larger, it's less likely to be toxic. And if it's smaller, it's more likely to be toxic. For skin permeability, as Pedro mentioned, the software will also take into consideration log P. So we know for agrochemicals in particular, more hydrophobic compounds, those that have low log P, maybe hydrophobic insecticides tend to be more active. So all of these factors are taken into consideration, and we will give you that likelihood level of, is it certain to be toxic? Is it plausible? Is it equivocal? Maybe there's some chance of it being toxic and some not, or is it impossible? So I think the best way to demonstrate software to you is to show you what it looks like. Now, Nexus is the application that we'll use, and Direct Nexus is a module as part of this. Within this same application, Sarah Nexus is also available, so you can use both those tools together, the statistical approach and the expert system. Here we have my compound that I have drawn in. You can also import or use SMILE codes. And we will just hover over to the blue Derek icon. When we click on this, we will get our prediction. If you're interested in the endpoints that we have available, you can go into the settings. And if I open these up, you can see all the potential endpoints that you can have. We also have some automatically calculated structure properties. So the log P, log Kp, and average molecular mass, again, to help inform the likelihood level. You can see on your screen now the prediction. It comes in just a few seconds. It does not take long at all. And here we have our summary of our results. So my compound here for chromosome damage in vitro, in mammals, this compound is plausible, and the alert that has fired is for an acridine or an analog. And I can see my area, the toxicophore that is responsible for this is highlighted. On this side of my screen, I have my comments, I have my examples, my results, and further information. So here, a description image defines the scope of the alert. So if you're developing a new active ingredient, if you're developing a new compound, you might want to see how you change your compound and how that will affect the activity. So you can see here, these R groups have to be a carbon or a nitrogen. If they're not, then the alert will not fire. So you can then tweak your compound in the software and see how that will affect the activity. And that will save you time, and it will also save you money because you don't need to synthesize your compounds and test them in AIMS. We have comments written by our scientists. Everything here is referenced. So they're not opinions, but they're facts. And you can see a full list of our references here as well. and you can click on them and go directly to the primary literature source. Again, helpful for when you're submitting to regulators, you know that you have all of the evidence. Finally, we also have some examples. So these are compounds that share the same toxicophore as your compound that you have put in, and these are essentially analogs. So this can be used for read across as well. You can have a look at your examples, and all of these have been tested as well. So for chromosome damage in hamster, this compound was tested in a chromosome aberration test, and the result was positive. We can see for mutagenicity in vitro, we might have slightly different examples, and they have been tested in AIMS in the Salmonella species, and the result was positive. Again, helping you with your read across and with your overall assessment of your compound. You can see for skin sensitization now, we are looking at a completely different section of our compound. So you can see the toxicophore is highlighted, and we have different comments, different references. You can use the software to analyze your compound, view all of the information that you might need, and you can also pull this information into a report, and you can see the direct nexus report on your screen. This is completely customizable, so you can add in sections for your comments. You can take away sections and add them to your weight of evidence assessment if you wanted to. You can see I have my submitted compound, my overall results. So I can see 4 endpoints have fired alerts, and they are plausible. I have my alert description with my comments, with all of my references as well. The final step in this would be expert review. So as Pedro also mentioned, you as the expert will need to go in and justify and review all of these results, because at the end of the day, this is a predictive tool. So we will provide you the prediction, but before you submit to regulators, you will need to be able to justify and just check that everything that you see in the software makes sense. Once you've done this, you can then submit to regulators. So that was a very brief summary that I wanted to show you just of the software. I know that we're a little bit over the time, but so just to conclude what I showed you today, in silico tools can be used to save time and money. It means that you're not developing your compounds, synthesizing your compounds. If you have multiple candidates, you can run them through the software, see if any of them fire genotox alerts, carcinogenicity alerts, and then narrow down your results. Our reports are accepted by regulatory authorities around the world, in Europe, in the USA. LASA have actually worked with regulators such as the FDA in the USA to ensure that our software is compliant. And not only this, but regulators also have access to our software. So when you submit reports via Direct Nexus, this is not an unknown quantity. Regulators will have seen these reports before, they know what to expect, so you can be confident in using this software. And as I mentioned, the reports are standard, but they're also customizable. So you can take the most relevant bits, you can add your own comments and your own expert review to the report, and then share this. If you're interested in more about LASA, our software, or how these tools can help you with your specific use case, whether that's in agro, pharma, cosmetics, please visit our website where you can find more information, not only on Direct Nexus, but also our other softwares, or you can get in touch with us via email. So I've included the email on your screen. It's just info@lasalimited.org. Or alternatively, you can also type in any questions, or if you're interested, put that into the chat, and I can get back to you after today's webinar. Thank you very much for listening.
Thank you, Raisa. Thank you very much. It was a great explanation about the LASA and the Derek system. And that was, I believe, for most of the people here, was something different and something new about what you showed us. So we have like, I put like some questions here in the chat to motivate someone to make some questions to us, right? But we have like more 5 minutes to answer any questions that you might have. So we are free here to Raisa or to Pedro answer to your questions. or even to webinar. So please feel free to write your question here in the chat. But anyway, if you don't have any other question here, we can you can send an email to us. You have a website that was shown during the presentation, and also our email that you can contact us. Right. So I thank you everybody personally to have you here, and in the next months we are going to show up the next webinar that we are going to promote. And we are going to let you know by LinkedIn and other platforms that communication platforms that Smart Talks, Bioacting, and LASA has. Okay, thank you very much. Have a nice Thursday, and have a nice weekend. Thank you very much, and see you next time.
Bye-bye. Thank you.
Thank you. Bye.
Bye-bye.